your entrances, because you are in the second half of the year already, right? It's July. And I hope you are not thinking of waking up when September ends, because that's very late then.
Sir, I have a doubt from the human reproduction chapter.
Yes, please ask.
So,
What were you waiting for all this time?
no, like I was just learning that chapter, and so in my teacher's notes, she gave for this placenta secretes some hormones, right?
Yeah. Can you see the whiteboard?
Yeah, and so there is HCG, HPL, progesterone, estrogen, and relaxin is produced by the secreted by the ovary. But then instead of progesterone, progesterone was also there, but she also gave progestogen.
Progestogen. Yeah. It's nothing but just anything. So let me tell you a rule for enzymes, and then you don't have to remember or mug it up. You have already studied in class 11th in digestive system an enzyme called pepsinogen. That converts to pepsin when the environment is acidic. You remember?
Yes, sir.
Remember pepsinogen, any enzyme which has gen in the name is just the inactive form or biologically not functional form of the enzyme. And every gen requires some factors, whether it is of environmental factor like the milieu around should be acidic, or it could be any other chemical factor, any molecular factor that converts it into the active enzyme that actually does the work. So what does pepsin do? What is pepsin basically? Remember what pepsin used to do? Pepsin, trypsin, chymotrypsin. Forgot everything. Fatima.
Yeah.
Remember what pepsin does?
Sir,
Where was pep?
Acts.
Yes, it acts where? It does acts. You're correct. It acts. But where does it act? Arpit.
so that was
Sir, it was to like, I'm not sure. I guess it was proteins.
Of course, it was produced, but where what it was produced and what does it do?
It digested proteins. I said proteins.
Proteins. Okay, you said it digests protein. Okay, and where was it produced from? Where do you think it is produced from? Remember gastric juices? There was something called gastric juices. In the stomach, there are gastric glands that secrete this. Yes or no?
Yes, sir. Yes, sir. Yes,
Yeah.
sir.
So basically, trypsin and pepsin are for, you're correct, they are for proteins, and amylases are for carbohydrates, right? Gastric amylase and pancreatic amylase. And pepsin and trypsin is for protein, then lipases is for fats. So here also the concept of gen is there. Now what Fatima's teacher told was what was it?
progestogen.
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 Progestogen. Yeah. So it becomes progesterone. Basically, progestogen, when it is secreted from the placenta, progesterone, the first contribution for progesterone during pregnancy comes from corpus luteum. Okay. And research says that the second contribution wave comes from the placenta itself, but it takes some time for the placenta to fully form and become functional. So the first kind of progesterone that it produces is called progestogen. Okay. There is another similar enzyme. Remember I told you it was present in one of those birth control methods. There was another form of synthetic progesterone which was present in oral pills. You remember that name? I told you there. So that is also another form of progesterone. So basically, progesterone is when it starts functioning to maintain the endometrium. So it's basically the same substance with just slight modification in its chemical formula. Is it clear, Fatima?
Yes, sir.
So this does not directly act to prevent the breakdown of endometrium, but this directly acts to maintain the endometrium and prevent the breakage. Okay. Similarly, you must also have read of two terms called estradiol and estrogen.
Yeah.
Yeah. Estradiol is actually form of estrogen only, where apart from just being the reproductive hormone, estradiol is also responsible for other things in the body, and estradiol is also present in males. It is present in all the males in the form. So estrogen is present in males in the form of estradiol, where it is responsible for some tissue development and especially brain development also during the early age. Okay.
Yes, sir.
Similarly, any other doubt you have? So what was basically that she told about progestogen? Any active role that your teacher talked about that you need a clarification in?
No,
Because
just about
progestogen is not important from reproductive or from the maintenance of endometrium point of view, but yes, it is secreted. Okay.
Yes, sir.
Yeah. Okay, very well. So let's start with today's class, and I think this is where we left in the last class. You all can see the screen that I'm sharing.
Yes, sir.
Okay. So can we go to Fatima today? Fatima, can you give me a summary today?
Yes, sir.
Yes, very good. Thank you so much. Let's start with the exceptions, which is linkage. Quick.
So first we did the statement for law of independent assortment, and then we like told about the exceptions of law of independent assortment, that is linkage between the genes. Linkage between the genes is like the functional physical association between two genes, which does not allow it to segregate, means separate independently. And then we talked about also, yeah, linkage is inversely proportional to recombination. So higher the number of higher the linkage, lesser the recombinations. And linkage was discovered by T.H. Morgan, and he studied it in Drosophila melanogaster, which is a fly. So what he did was he took a yellow-bodied and white-eyed female Drosophila melanogaster, and he crossed it with a brown-bodied and red-eyed male Drosophila, and then he got the F1 generation, and he got the F2 generation by selfing, and he saw that the phenotypic ratio wasn't the same as the phenotypic ratio Mendel's got in the pea plant. So he said that the number of recombinants he found out that the number of recombinants were very low, and recombinants means non-parental type. And then we discussed about the chromosomal theory of inheritance, which like the whole work was done by Mendel's, but like the people, like the two scientists who stated it was Sutton and Bovary, and they did it in 1900. Before that, we discussed some few points, and that was like Mendel's work got published around 1865. It was not accepted till 1900s, and the people, the scientists, I'm so sorry, the scientists who gave the
scientists are people only. No
yeah,
problem.
but it's rude to call them that, so
you can call them uncles also, it's fine. Okay.
Yeah, so the scientists who gave the three laws were Hugo de Vries, von Schirmack, I don't know how to pronounce them, and Carl Correns. These three scientists were the scientists who gave the laws. Then,
So
yeah,
basically, if it comes in any entrance question, remember the major one is Hugo de Vries, who actually raised the status to laws. Okay.
yeah.
The other two were also there and independently rediscovered the work, and also they also agreed that yes, what Mendel did was logical and it should be a law. Yeah.
Okay, then the chromosomal theory of inheritance, in which this was the because of this theory, Mendel's work was validated.
Yeah,
And
but okay, before we go there, because we talked about T.H. Morgan, any one of you know what is a Drosophila? Have you seen a Drosophila in
it's
reality? Anyone has seen a Drosophila in reality? So I was going through some previous research work of mine, and I actually found out some pictures, interesting pictures that I took of the Drosophila. So this is the fly. Can you see the screen that I'm sharing?
yes, sir.
Yeah. So this is a fruit fly, a Drosophila, and luckily this fruit fly is laying egg before it was anesthetized. So basically this fly is unconscious and it was in the process of giving an egg. So the egg can still be seen here. And can you see this red eye phenotype, which we talked
Yes.
about? So this is the red eye, which is always linked to the brown body. Can you see the body is brown?
Yes, sir.
This is the body. Brown body and red eyed is a linked gene. I do not have a picture for red, white eyed and yellow bodied, but I think that's okay. Yes, continue, please. I also
Sir,
have some
you took
larvae.
that picture.
This is just one moment. Wait. Let me open the folder. Have you seen Drosophila larvae? Anyone? It's very interesting to see how they develop. I have, yes, I think I can. I hope I can share it. Can you all see something?
No, sir.
Can you see this?
Yeah, it's visible
Yes,
now.
sir.
This is all right. Let's see if. What can I see? Can you see this bottle?
No.
No. Okay, that's the. Okay, so I have to share it like this. Okay, let me do one thing. Show you some pupae.
Sir,
Yes, tell me.
sir, like why it is not opening?
No, it's opening. It's open. Don't worry. Yes, because I have to like in Zoom you have to share everything separately. Can you see this transition?
Yeah. Yes, sir.
So it starts. So this is like one pupae. So Drosophila gives larvae. This is a larva. Can you see at the very bottom where my cursor is hovering? So this is a larva. Okay, and this larva eats a lot, keeps eating. It comes out of that egg which you are seeing coming out of the abdomen of the fly. And then this larva stops and fixes itself. And it fixes, and there are two tentacle-like things that come like horns. That is the head region where it comes as horns. And then over time it starts getting yellowish. And within this is the outer skin that gets detached from the inner skin, and inside a fly is developing. The same thing happens for butterfly, cocoon, and every other insect that do metamorphosis. So from this pupa, after after like 6-7 days, the fly will come out. So this was just an overnight observation that I did for Drosophila larvae and pupae. Interesting, right? And the picture that you were talking about, Just one moment. No, this is not my picture, but
So your
I must be.
screen is not shared.
Yeah, I'm not. I have not shared it actually. I was thinking if I have a Drosophila picture, live Drosophila, but I have larvae and pupae, most of it. Yeah. Oh no, I have Drosophila picture. I have video also. Would you like to see a video?
Yes.
Yes, sir. Yes, sir.
Great. Let's see what's the video about. Let me see what it is about. Oh, that's about Drosophila mating, reproduction. We can connect it to there. Good. Let me. You would like to see that, right? This is just an old folder that I found, and it's interesting actually. Can you see the screen, people?
Yes.
Everyone can see the screen.
Yes.
Yes, sir.
So this is on the opposite side. So the picture is taken like basically it's inverted. So I'm imaging from the downside. You know, they are on the Petri plate, which is transparent. So they are on a glass. Can you see this red eye phenotype here also?
Yes,
This red
sir.
eye with brown body. Yes. So
Yes,
this
sir.
is a female. Is this thing coming in between? Oh yes. Yes. Now this is a female. Can you see females are bigger in size than males? This is
Yes,
a
sir.
male. And this female is on a glass, and I'm looking from the downside. Do you understand? That's why the belly is visible. These are the legs, red eye, and the
Oh,
body.
yes, sir. Got it.
But don't
And
you have
it
to see through microscope?
this is through microscope only.
So why is it down?
So can't you put it on the eyepiece from where we see the?
Yeah.
Yes. So this is the eyepiece. You know, I'm recording this through a mobile camera, just for my curiosity. So, and from the down, you see like this is where the Drosophila is, and this is the male. So male actually is mating with the Drosophila. It's depositing the sperms in the female spermatheca. So now can you see that how they are walking? I am just looking from the downside. Can you see this?
Yes, sir.
Yes. So this male is trying to reproduce with this female, and there is another male, you know, because probably between these two, he won and he lost. So the male who wins over the female, or who impresses the female, gets to reproduce with the female. Okay. And this is a behavior of flies, not even Drosophila. This is the fruit fly, but also in other flies, you will see this behavior. Can you see this fly? They constantly keep rubbing their front hands, cleaning it, then cleaning the eye also. Can you see this?
Yes, sir.
This fly? Yeah. So this is what flies constantly keep doing, if you observe the fly. These are Drosophila. They are very, very beautiful model organisms to study genetics, because they have such contrasting phenotypes. They are small, and they have even smaller life cycle than a pea plant. And Morgan studied a lot of interesting things here. So the lab I used to work during my master's research worked on Drosophila. I also worked on zebrafish. Someday when we will be talking about fish or something, I will show you zebrafish. Okay. And I have something.
Sir, did you have to touch the flies?
Did I have to
Touch the flies.
dust the flies? Means,
Touch.
of course,
Yeah.
you cannot work without touching them, no? So we used to just give me 1 second. Let me see if I can show you something more interesting. It is there. Now I'll show you in the next class. I'll show you Drosophila brain. The whole brain taken out, a very small, tiny, microscopic brain imaged in under a microscope. Yes, now come up with your questions. What you asked, do we have to touch the flies? Yes, we
Yeah.
do have to touch the flies because we have to like breed them, crossbreed them, mutate them, cause mutations, and in genetics you see that in next generation which trait is going, what you want to study. So basically the way you touch them is you keep them in bottles with media, like food which they eat. It's corn meal, it's made up of corn flour, and you can just make them unconscious. So they get unconscious if they experience sudden cold temperatures or CO2, then they are gone. And then you can sort them, touch them, do whatever you want, and then put them back in the bottle where they will come out of their unconscious state, and again they will start eating and staying there. So this way they do not fly away. Yes, continue, Fatima, with the. So I just wanted you to show Drosophila. Drosophila is all around you, okay? If
Were
you
you wearing gloves when you were touching them?
Yes, of course. Yes. You have to wear gloves whenever you do any kind of molecular biology. For your safety, like you know that even if the flies are not disease, but you can contaminate the fly because your hands are sweaty all the time. You may have pathogens and dust and everything on your hands. So you need to wear gloves. For the same purpose, like doctors, when you will be doctors, you will be wearing gloves when you will be examining the patient or doing a surgery or something to keep it uncontaminated. Okay, let's come to Sutton and Boveri now.
Yeah. So Sutton and Boveri gave a theory which stated that the pairing and separation of a pair of chromosomes would lead to the pairing and segregation of a pair of factors that they carry.
Yes. So
Okay.
basically, Sutton and Boveri just legitimized Mendel's work by saying that what Mendel's kept saying as factors, they are basically genes present on chromosomes. And you can see that whenever you separate a chromosome pair, the genes, the traits also get separated, right?
Yes.
Yes.
So, and then we discussed about recombination, like a small discussion. Like, so we said that recombination is the production of non-parental type of gene combinations through meiosis.
Yes. And
And
just add one more line here. It happens through, like when it happens, it happens through physical breaking apart and rejoining of chromatids. Okay, the thing
But
that I told you about.
this we aren't doing, no, it's happening like that only.
It's happening. Yes,
Okay.
it's physically breaking and then rejoining on its own through crossing over.
And
Yeah.
then we discussed about homologous, some basic terms, homologous chromosomes, then chromatids, sister chromatids, non-sister chromatids, crossing over. So, do you want me to
What
tell
are-
this in detail?
Yeah, I wanted to tell you what are homologous chromosomes and sister and non-sister chromatids. Just like what you see on the screen. Just explain what is a chromosome. That's all.
Okay. So, homologous chromosomes are chromosomes which have same length, and the length of each arm is also same, and the genes are located at the same locus, and same
Yes.
genes are also there. Okay, that's
Yes.
homologous chromosomes.
What are sister chromatids?
The chromatids are, the Y and the I don't know how to explain it, but I know what it is.
So they are, yes. So they are the two chromatids of the same chromosome, which are exactly the photocopies of each other.
Yeah.
Okay.
And non-sister chromatids are chromatids of different chromosomes.
Non-sister chromatids are chromatids of two different but homologous chromosomes.
Homologous chromosomes, yeah. And then we did crossing over.
Yeah.
And then we did polygenic inheritance. So polygenic inheritance is like one character is controlled by more than two genes. It's called polygenic inheritance.
Not, yes, more than two genes.
Yeah, more than two genes. Example for it is human height and human skin color.
Yeah.
And it's a physical like environment can also affect these two
Correct.
characters. Okay, then we did pleiotropy. Pleiotropy is where a single gene is controlling many characters, more than two characters,
Yeah.
many characters. Um, and then for that example was phenylketonuria, in which a single gene gets mutated, and that single gene codes for different enzymes. No, that this single gene codes for phenylalanine hydroxylase, but the many symptoms are like we saw many symptoms in the person who has phenylketonuria, and symptoms were mental retardation, then reduced pigmentation of skin, reduced pigmentation of hair, and
Yes.
by this we concluded that one gene is responsible for many characters.
Yes, perfect. Thank you so much, Fatima, for the brief recap. Hello, Alvia. You just joined in. How are you? Am I audible to you? Because you are not audible to me. Yes, I could hear some sound.
Hello.
Yes, you're audible now. You missed quite a few classes. Where were you?
So actually it's like raining here and there's like some bad weather problems.
Okay. Where are you
So my
currently?
Wi-Fi was not working. Hello?
Yes, where are you currently?
Oh, I'm in Mumbai.
You are in Mumbai. I can understand. Mumbai and Pune are, they have very bad weather and connectivity right now. Okay, anyway, so today we will be starting with sex determination, and also we will be starting with genetic disorders. Okay, Alvia. So I would like to start with a new page here. Okay, everyone. So let's start with today's topics. The first thing that we will do is in genetics, like genetic basis of sex determination. So as you already know about humans, we have discussed about human sex determination multiple times in chapter 3 and then again in chapter 4 when we were doing amniocentesis, right? So quickly about human, it's just a revision. How do you think human sex is decided or determined? In humans, there are two sex chromosomes. One is X chromosome and the other is Y chromosome. And these two are called sex chromosomes. These two chromosomes, when they pair, decide the sex of the human baby. If it is a female, then in the female it will be X and X, right? And if it is X and Y, it is a male, correct? Now in humans, you already know that females are homogametic. Yes or no? You know what is homogametic? They give rise to only one type of gametes. Is it clear? So their gametes contain 22 autosomes and one X. And all their gametes contain that because they have two X. Whereas males in humans are heterogametic. We have already discussed this, correct? Just revising this. And there are two different gametes possible. Everyone understands how? Samira, Arpit, Kulsum, Fatima?
Yes.
Yes, you understand how? This is heterogametic and this is homogametic? because when they will
Yes.
form gamete, it will be 22 autosomes plus X or 22 autosomes plus Y. There can be two different type of gametes. In females, it will be all the gametes will be same. So in humans, it is XY system. So this system in humans is known as XY system of sex determination. Just write this down. XY system. Now the same XY system apart from humans is also in Drosophila. Okay, is it clear, everyone?
Okay. Yes, sir.
Yeah. Now in Drosophila also, males are heterogametic like humans, and females are homogametic. Okay, in fruit fly. The second, so this is the first type that is there in your syllabus. The second type is called X naught system. X naught system. Now in this kind of system, basically it is the number of X chromosomes that decide the sex, because there is no Y chromosome, there is no second sex chromosome. So write down, in this system only one sex chromosome is there, which is X. Only one sex chromosome is there, and that is X chromosome. And if it is X and X, which is 2X, a couple of X chromosomes, then it will become a female. Yes. And if it is X, just one single X with nothing, so this O is not, it is like the O blood group, there is no antigen. If it is X naught, then it becomes a male. Is it clear, everyone? So if there is only one copy of X, it becomes a male, and if there is two copies of X, it becomes a female. And this type happens in cockroaches. Yes, cockroaches, grasshoppers are the example. Is it clear? So in cockroaches and grasshoppers also, females are homogametic, right? What are males? They only have one type of gametes, so they are homogametic, and males are heterogametic. Do you understand? Because in one of the gamete they will have a X pair, in the other they will have nothing. You understand, everyone?
Yes, sir.
So in both XY or XO system, females are homogametic and males are heterogametic. That you have to remember. Drosophila, cockroach, grasshoppers, and humans all have heterogametic females. Now this was a neat question asked in 2015. Written this thing, noted down. Yes or no? Noted down.
Yes, sir.
Yeah. Now the third system is called ZW. In this, there is no X or Y chromosome. Instead, there is a Z and a W chromosome. Now the different notation of the chromosome is given because they are different types of chromosome. They are not like X and Y chromosomes, but they are responsible for sex determination. Now in this type, there is a difference. In this, males are Z and Z. Okay, so this is male. So males becomes
Homogametic.
Yes. And females are Z and W. One way to remember how I remember it is W for woman. So males are homogametic. They produce the same kind of gametes, and females are heterogametic. And this ZW system is present in
Birds.
yes, birds. Specifically, example is chicken. Okay, most of the birds, not all birds, but most of the birds. Specific example is chicken. So in birds or in chicken, who's responsible for the sex of the baby? Father or the mother?
Female.
The mother. Okay. Is the ZW system clear to you? Now all these systems are on the basis of male heterogamety or female heterogamety, but all these were chromosome based, so they were very simple. All, whether it is cockroach, grasshopper, exo system, ZW system, or XY system, all are chromosome based system where there is heterogamety or homogamety. A very interesting one, which is favorite for entrance questions, is sex determination in honeybees. Now NCERT talks about honeybees specifically, but the same kind of sex determination also works for ants and wasps. Okay, if it is asked in entrance, because honeybees are closest to ants and wasps. Okay, they all basically sting. This is how you can remember. But in honeybees, it is a little interesting. Now, before going into the sex determination, let me tell you something about the honeybee. So, honeybees are social animals, just like ants. They are called social organisms. You know why they are called social organisms? Because they live in colonies. They never live alone, solitary. They live in colonies, correct? And what is the colony of honeybees known as?
Honeycomb.
Yes, honeycomb or beehive, right? Beehive. Now, in a beehive, there is a division of labor in the society. In honeybee society, your work is decided right from the birth. So whether you are an engineer, a doctor, a scientist, or whoever, whatever, if humans were to live like that, right from the birth, it will be decided. So a division of labor is present by birth. So everyone knows their role right from the birth. Now they cannot change it after the birth. Okay. In a beehive, there are 3 types of bees. Anyone knows about those? And this is important to understand. This is not in NCERT, but if you don't understand this, sex determination will be slightly difficult to understand. So there are 3 types of bees. First one is a queen bee. You must have heard. What? What, Arpit?
I think so.
Sorry, I could not understand. Come again.
Sir, I was thinking there are also worker bees.
Yes. So first tell me about queen bee. What is a queen bee? Queen bee is a male or a female?
Female, probably.
Yes. It is the only fertile female. Okay. Capable of laying eggs. So technically, all the eggs which are laid in a beehive are laid by the queen bee. That's why a bee colony cannot survive without a queen bee. And you must have heard or read or seen, if you take a queen bee, whenever you have to establish a bee colony, you don't have to worry about all the bees. You don't have to collect thousands of bees and put it somewhere. Just kidnap their queen bee and put it somewhere. and that queen bee will keep producing pheromones, and all the workers and other bees will come to that area, that place where queen bee is, and establish a new colony. Okay.
Sir,
Yes.
so how can we differentiate like which is the queen bee?
Queen bee is the biggest bee. So morphologically, it's the biggest, and you can differentiate it very easily in any colony. It is the biggest bee, and it lays all the eggs. Okay.
Okay,
And
sir.
it's mostly protected. It always remain covered by all the bees, and it is towards the center of the beehive. It's not like at the periphery, because if a bird attacks the beehive, like other bees can sacrifice themselves or can die in fighting, but queen bee should not be killed, because if queen bee is killed, then the whole order of that beehive will be lost, and workers and drone bees will become chaotic and will just die. They will not know what to do. So all the maintenance, the signal to the commands come from queen bee as chemical pheromones. So queen bee is the only fertile female capable of laying eggs. And apart from queen bee, there are worker bees. You know what are worker bees? Their sex?
They are infertile.
Yes, they are also females, but they are infertile females. Okay. They are infertile females. And they are responsible for making the colony, expanding the beehive, cleaning the beehive, bringing the nectar, depositing the nectar, everything is done by worker bees. That's why they are called worker. They are the workers of the hive. And you know, how are they fertile? That's also very interesting in biology. How all the worker bees are fertile. Where does queen bee comes from? You know that? How is queen bee produced? Queen bee is chosen. So every queen bee is actually chosen from a pool of larvae, which are female larvae. And one of that larva is chosen and fed by royal jelly. Royal jelly is a very nutritious thing, which is stored in the queen bee chamber. And if any larva feeds on that, then that bee will grow to become queen bee. Queen bee secretes some pheromones like chemical messengers. If the chemical messengers are working inside the body, they are called hormones. If the chemical messengers are working outside the body, they are called pheromones. Is it clear?
So can you explain again?
So, you know, hormones are also chemical messengers, but they work within the body.
Yes, sir.
Inside the body, between organs. If an organism secretes something, a chemical, which gives signal to other organisms, or which causes some change in the behavior of other organisms, that's called pheromone. So that's a chemical messenger that works outside the body. Is it clear?
Yes, sir.
So worker bee secretes a pheromone that keeps queen bee secretes a pheromone that keeps the worker bees unfertile. It does not allow their reproductive organs to develop. So it's like a very cruel but control and regulation that is kept by the queen bee so that no other bee can take her place. Okay. And there should be only one queen bee in one hive. So worker bees remain unfertile under the effect
Sir?
of queen bee. Yes.
So there is no king.
There's no king. There's no concept of kings in bee colony. It's the female world, female dominated matriarchal world. Yes, Fatima, so much of politics. Not politics, but you know, it's so much of biology to keep a control on a colony. You know, so it's biological politics. So queen bee is not chosen, you know, she's just there. Until the time she's there, she's the leader of the hive. And you cannot democratically vote out the queen bee and make someone else the queen bee. When the queen bee dies, or when she's about to die, you know, another queen bee is chosen before the queen bee dies from the pool of queen bee.
Oh.
You know, that's what I said. There's no politics. There's no democracy, voting or something like that.
Hierarchical system is there.
Yes, it's like queen, and the one the queen chooses, the larva that feeds on royal jelly becomes the next queen, and so on. The hive continues. If the queen bee is not there, then the workers will not be under the effect of that regulation. They will not be disciplined. They will become chaotic, and they will start becoming looking for reproducing mates, and they will not look after the hive, and everything will scatter. And because bees cannot survive alone, so everyone will die. Okay, so rebellion here means death. So if you want to live peacefully with the queen. Okay. Infertile females are worker bees. Then the third kind of bees are drones. Drone bees. And drone bees are fertile males. So there are drones and there are queen. Workers are infertile. So all the drones of the hive, they can only reproduce with queen bee. So drone bees, basically, just like I showed you Drosophila. Remember that male was trying to deposit his sperms in the female spermatheca. So these insects, the females have a pouch, a special pouch in which they can store the sperm of the male. So drone bees actually reproduce with queen bees and put their sperm in their spermatheca. Is it clear?
Yes, sir.
Theca means bag. Yes, Arpit.
The drone bees are like indirectly kings. Like
No, they are not kings. They are like Pehredar. I'll tell you why they are not kings.
okay,
You know, there is no king, as I told you, because they are very, very small. They just go turn by turn when the queen allows, and just deposit the spermatheca. And they are not kings because kings are essential. You know, a king is an essential entity. It is not something, you know, just nominal. But drones are not essential. Queen bee can reproduce without drones, without sperms. Okay, so that is also, that's why I said that they are not kings. So did you understand this hierarchy?
yes, sir.
Yes, sir.
Now let's come to how their sex is determined. So the system of sex determination in honeybees is not dependent on any one particular type of chromosome or the sex chromosomes. It is dependent on the copy number of the whole genome, which is called ploidy. So this system is called Haplodeploidy system. Haplodeploidy system of sex determination. Okay. Now let me explain you what is this haplodeploidy system. Now pay attention here. I'm starting from a female. I'm starting from a queen. Okay. From a female, which automatically will be a queen because only queen can reproduce, right? So look at the first case here. A queen, which is a female. All the females, let me use some other color here. Queens are females and workers are females, right? Both of these are females and their genome is diploid. Is it clear?
Yes, sir.
So they are diploid organisms, and all the males in the bee are haploid. Their body is haploid. It's not like their gametes are haploid, their body is haploid. Like in our cell, in each and every cell apart from the gametes, we have two copies of the genome, right? One from the father, one from the mother.
Yes, sir.
But in bees, all the males have only one copy. They are haploid, and all the females are diploid. So both queen bee and worker bees are diploid, but only queen bee is fertile. So I'll start with queen bee here. So if there is a queen bee, she is diploid, then diploid means 32 chromosomes. 32 chromosomes in honeybees is the 2n number. Is it clear? So what is haploid number?
16.
16. What is the haploid number in humans?
23.
Perfect. Now, if a queen wants to make gametes, okay, so gametogenesis in queen will happen through mitosis or meiosis?
Meiosis.
Yes, because it's a diploid organism, and diploid organisms can only undergo meiosis, right? Haploid organisms cannot do meiosis. Remember, I told you very early in chapter 3.
Yes, sir.
Yes. So when they form gametes, they form it through meiosis. So their gamete will be eggs. So let's say this is one egg, and this is another egg. Okay. Now what will be the ploidy of the egg? 16 chromosomes, which is the N number,
N.
right?
Yes, sir.
16 chromosomes, which is the N number. Remember, I told you very early that N and 2N is just the number copy. So see, 16 is N for honeybees, but for humans, the N is 23. Okay. Now the eggs have two ways it can go. One way is this egg fuses with a sperm. Okay, sperm will be produced by males, of course. And if this is 16, then sperm will also be N, right? And when they both fuses together, they give rise to a diploid organism, which will be a female. So females are diploid, and all the females, sorry, write down, through sexual reproduction, in a honeybee, which is fusion of male and female gamete. Only females are produced. So if there is sexual reproduction, only females will be produced, because only females are diploid. Is it clear?
So
The other way is, this is the egg, and the egg remains unfertilized. I told you, queen bee can also work without any sperm or males. So if it remains unfertilized, which means there was no male in the colony, right, to deposit sperms.
Yes, sir.
And if it remains unfertilized, then the queen undergoes a process called parthenogenesis. You have studied parthenocarpy, right? Remember parthenocarpic fruits in plants? Remember, yes or no? And through parthenogenesis, only males are produced. Because if it is unfertilized, the chromosome number will remain haploid, and all the males are haploid. So, and what is parthenogenesis? You can write down. What is a parthenogenesis? No. Development of embryo without fertilization. Is it clear how are males produced and how are females produced?
Yes, sir.
So if there is fertilization, there will be all females produced. If there is no fertilization, only parthenogenesis, then there will be male produced.
Sir,
That's why, yes, Fatima.
does the female mate with its own children?
Yes. There's no concept of children or parents socially in animals, you know. Apart from humans and some high order primates like chimpanzees and orangutans and monkeys, and also elephants to some extent, the concept of son, daughter, mother and father does not exist because they do not live like a family. Okay, so you know that the moment a lion, so in a pride of lion or in a group of tigers, there is always an alpha male, you know, the male who actually protects the tribe, who's the fittest and the strongest male, and he reproduces with all the females, right? You know that? That's the alpha male. If anyone has to challenge the alpha male, then they have to defeat the alpha male or kill the alpha male or cast out the alpha male from the pride. And then the male who actually do that, most of the time, it is that alpha male's son only. Most of the times. That's why whenever a alpha male takes over a new pride, like suppose there was a male roaming solitary and he saw a pride, a group of lion with, like, there were 4 lioness and there is 1 alpha male and 2-3 cubs. So he will attack that alpha male. If he defeats that alpha male, the first thing that he does behaviorally is kill all the kids of that alpha male. You understand why?
Yes, sir.
Because otherwise that kid will grow up and will challenge him. And when he will challenge him and will become the alpha male, the kid will, you know, reproduce with his own mother if she's in a reproductive age by then. You understand? Biological mother. So there's no concept. So that's why most of the times whenever the and there's also this thing, whenever a kid starts growing up and becoming an adult, he leaves the pride. He leaves his mother's and the father. Otherwise, he will be a contingent, like he will be one of the competitors of his father and he has to fight. So he leaves and then he tries go and find a pride or take over a pride. So this is a very common behavior among wildcats. Same is for like flies or honeybees. They have no concept. Like we know that biologically who's parent and who's son or daughter, but they don't know that, right? They do not name their kids, oh, you are my daughter. They don't know it. Yeah. Okay, cool. So is this clear about
Yes.
if we start from female? On the other hand, if I start from a male, which will be a drone, right? So male or a drone. So male is already to begin with a haploid, right? It has 16 chromosomes and it is haploid. Now, if a male has to produce gametes, sperms cannot be half. They have to be haploid. Body is already haploid. So gametogenesis will happen through mitosis here. Mitosis.
Sorry.
Okay. So mitosis in honeybees, that's the difference. We always think that meiosis give rise to gametes, but in honeybees, male mitosis can give rise to gametes because their body is already haploid. So through mitosis, they give rise to sperms, and these sperms are also N. Now these sperms have only one option, to fuse with the egg. Yes or no?
Yes.
And as a result of this, what will be produced?
Female.
A female. All the time, whenever a male is reproducing, a male can only reproduce through sexual reproduction, and a male can only give rise to females or daughters. So a male honeybee can never give rise to sons. Interesting. A male honeybee has no son. They do not have sons and they do not have fathers. Do you understand? This is the neat question. That's why I'm explaining you through this. A male honeybee never had a father and never will have a son. You understand why? Because males come from unfertilized eggs, so they only have a mother, and males can only reproduce through sexual reproduction, so they can only give rise to a daughter, a female. You understand, everyone?
Yes, sir.
Yes, sir.
A little complicated, but yes. Arpit.
Can you scroll up, sir? Written, sir.
Yeah, I will. Please let me know when you are done writing. Then I'll tell you some interesting questions that have come.
Sir,
Yes, Arpit.
sir, I'm actually having a doubt in the sir,
Mhm.
ZW system. Sir, like why do we differentiated it, sir? Like in the humans male, it is homogametic XX and it's like very similar. So like what is the need of differentiation?
In ZW, females are heterogametic. Females are ZW. Males are ZZ. In humans, males are XY heterogametic and females are XX. So that was the need to differentiate. Because
Okay.
in birds, it's female heterogam. Yes, in birds, it is female heterogamety that occurs. In humans and in Drosophila, it is male heterogamety that occurs. Whereas in cockroaches also, it is the male heterogamety that occurs. Okay.
Yes,
So that was the
got
diff. Yes. This is drawn. This is done. So remember that haplodeploidy system, because it's something new and can have many questions on it. So this is asked the most. And also remember that first point, write down, male honeybees do not have fathers and sons. Okay. Now, let me ask you a question. Do male honeybees have grandfathers and grandsons? If they do not have fathers and sons, can they have grandfathers and grandsons?
Yes,
Yes, Fatima, how?
because
Because
grandfather, because the mother, what male, the male,
Yes, sir. Yes, sir. They can have.
yes, that is
male
what I'm asking.
can
Yes.
be male can be produced by a female, okay,
Yes.
and female can have a father.
Yes.
So
So
that's
males
grand-
have mothers and their mothers have fathers. So males can have grandfathers that way.
Yeah, that's grandfather.
And
Okay,
males
that's-
have daughters and their daughters can produce sons.
Yeah.
So males can have grandsons, but not fathers and sons. So write down.
Sir, do they really ask these relationships in exams?
Yeah, exactly. They will, because often in NCRT also no one talks about the relationship. They only tell you about that, there is haplodeploidy. N is male, and I could have just finished in 30 seconds. But why I'm spending 10-15 minutes on this topic? Because you need to understand every detail of it. These details are important because just knowing that 16 N is male and 32, which is 2N, is female is not enough, right?
Yes, sir.
So males honeybees can have grandfathers and grandsons. There is no such rule for female. Female can have everything. Daughter, son, granddaughter, grandson, grandfathers, grandmothers, everything. Okay. Just applies to males.
Can you make us write the reason also?
Can I do what?
The reason.
The reason for?
They can have grandfathers and grandsons and not fathers.
Okay, yes, please. I will do that. So as I told you, males are haploid, right? Males are produced from unfertilized egg cell. So when the egg is unfertilized, a male is produced, okay, through parthenogenesis. This is clear?
Yes, sir.
So if it is produced from unfertilized parthenogenesis, it never had a father, right? There was no father to contribute sperm.
No.
A male only comes out of a egg cell. So that's why a male remains haploid. Is it clear?
Okay,
So
sir.
this is how males cannot have fathers, but they can have grandfathers because the mother from which they were formed, from whose egg they were formed, that mother had a father, right? Because females are produced through sexual reproduction. Both father and mother is needed. So males cannot have fathers, but they can have grandfathers. Okay. Similarly, a male's only option when it produces sperms is to undergo sexual reproduction with a female, which most of the time is the queen. And a male can only sexually reproduce to give rise to a daughter, a female, because through sexual reproduction only females come up. So that's how a male can only have a daughter, not a son. Okay, is this clear? Yes. And this daughter can further go on to give son and daughters both. So the male can have a grandson through this daughter, but he cannot have a son himself. Is it clear? So they have grandsons and grandfathers, but they do not have fathers and sons. Is it clear?
Yes, sir. It's clear.
Okay, very good. Shall I move forward, everyone? Everyone has noted everything? Say yes or no?
Yes, sir.
Yes, sir.
Okay. So let's start with the next concept. And this concept is very important one, both for all, like related to all the chapters that you have studied till now. And that is called mutation. What is happening? Yes, mutation. Anyone knows what is a mutation? Now is the time to speak.
Sudden change in the behavior.
Sudden change in the is not a complete answer.
Change in
Yes.
DNA sequence.
Change in DNA sequence? How? How can a DNA sequence be changed?
Sir, the genes can be changed.
How? You are just saying what else you said.
Alteration.
Yes, sudden changes are called alterations. Like you had to do alterations to your,
Deletions,
Eid dress.
insertions.
Yes.
Yes.
So there can be issues happening with the DNA. For example, there can be a deletion at any one point, right? Some part of the DNA got deleted through a mistake. Something extra got added, or something got mutated to become different things. So I remember I told you there are 4 types of bases in the DNA: A, T, G, and C. Adenine,
Yes.
thymine, guanosine, cytosine. They are 4 nitrogenous bases. 2 of them are called purines, which is A and G, and 2 are pyrimidines, which is T and C. And they all come, like one purine combines with one pyrimidine, and that is how a ladder-like structure of DNA is formed. I'll not go back. You can go back and revise. I have done it in the beginning of sexual reproduction, right? We studied DNA, chromosome, everything, right? Gene, etc. That's why I did it there. Is it clear, everyone? So mutations are the sudden changes due to deletion, insertion, or or alteration in the sequence of the DNA. And do you think mutations can be carried to the next generation? What do you think? Yes or no? Can a
Sir,
mutation? Yeah.
not necessarily, but yes, they can be.
If not necessarily, then how? Yes. Why yes? Under what condition? Yes.
Because they can be inherited.
How? If they can be inherited, why not all mutations are inherited?
They can be inherited.
All mutations can be inherited?
Yes, sir.
No. Only those mutations will be inherited which affects the gametes. Okay? Or the gonads. Because whatever is in the gamete will only go to the next generation. Whatever is not in the gamete will not go to the next generation. For example, you learned a language. For example, you know French. Now, French, the capability of you knowing French is not in your genes. It is an acquired character, right? You learned it. So there is no gene for French, knowing French. For that matter, there is no gene for any language. So the kid will not know any language by birth, right? He has to learn a language. Do you understand that? So anything that for and in case of mutations, let me tell you give you another example. You know that our skin can get mutated, DNA damage can happen in the effect of UV radiation. If someone is exposed to ultraviolet radiation, their skin cells can have DNA damage or mutations in the DNA. Yes?
Yes, sir.
Now, those mutations, if they are just localized or they are restricted to the skin cells only, will not pass on to the next generation. So there is a mutation in an organism, but that mutation did not enter the gametes. So it will not be passed on to the next generation. Do you understand, everyone?
Yes, sir.
Yes. So write down mutation. Let's define it. Write down. It is the sudden
mutations harmful to the organism.
Yes, mutations are mostly harmful to the organisms. The chances of some mutation being useful is very, very less. Okay, because see, already you have a genome. It's a very good question, Miraj. Already you have a working blueprint, right? It was working fine. You have a DNA which is doing everything for an organism, which means it is a good piece of DNA working well, right, Miraj?
Yes, sir.
Now something randomly got changed. Now, if you change something randomly, it's like there is a painting, there is a already made beautiful scenery, and I gave you a color palette and I blindfold you. I close your eyes and you touch the paintbrush in any of the paint. You don't know what you are doing because mutations are just random and sudden. And you touch that color paintbrush anywhere on the painting. So out of 100 times, 98% or 99% of the times you will just ruin the painting, right? You will not add something to it. You understand? Because everything is in order.
Yes, sir.
There is a sun, there are some trees you have made, there is a river that you have made beautiful, and some animals are there. Maybe when you touch that painting randomly with any color, that any color could be black and it could just make a spot on the sun or anywhere. So it will just ruin the painting. So the chances of it damaging what already is functional is more, right?
Yes, sir.
But it can also happen by chance that you just dipped your paintbrush in a green paint and you just touched at a point which was a tree, and that shade of green just, add a beauty point to that tree. That's also possible.
Yes, sir.
But that's a lucky chance event, like a lucky draw. So in a similar manner, some mutations can be helpful to that organism. And mutations that are helpful, only those are passed on, because those which are harmful, they will just kill the organism only, right? So no organism, no formation of gametes, no reproduction, no passing on of the mutation. So if there is a lethal mutation, a detrimental mutation, it will just end with that organism. So we never know about how many mutations were lethal because we cannot study it, because no organism survives to pass it on. But some mutations gets passed on even if they are detrimental. And most of the times mutations which are beneficial are accepted over time. But this is a very slow process. You understand, Miraj?
Yes.
Only once or twice in a generation, there can be a mutation by chance that can be of any help to any organism. Not more than that. Yes, so write down. Let's first define it. Write down. Mutations are sudden random alterations. They are sudden and random alterations in the DNA sequence. In the DNA sequence that consequently, as a consequence, that consequently results in the change in genotype and phenotype. Because if there is a change in the genotype, there will be a change in the phenotype as well, correct? Of an organism. Is this part clear? What are
Yes,
mutations?
sir.
Now let me come to one more part. Remember when we were studying asexual reproduction versus sexual reproduction, there was a point. So I told you, whenever we study any science, there is a way to study it. You first read the fundamentals, then as you keep going deeper into the details, you realize that some things that you studied were not 100% correct, and you again come back, unlearn and relearn. So you all told me, it does not mean that what you told me as an answer is incorrect. That's correct. You all told me that asexual reproduction does not give rise to any variations, right? Asexually reproducing organisms are clones of their parents, right? Remember these statements, you only made it.
Yes, sir.
Sexually reproducing organisms give rise to variations, and they are not clones. They are not identical to their parents. So let me ask you a question now. Bacteria are asexually reproducing or sexually reproducing?
Asexual.
Asexual.
Very good. So technically all the bacteria belonging to one species in this world are all clones of each other? Yes? Yes, technically. They should be, right, Fatima?
Yeah, they should be.
Yes. Then why is it that we could kill the same bacteria 30 years ago with an antibiotic, but today in 2022, we are not able to kill that same bacteria with the same antibiotic with the same effect? It's not effective now. And we call those bacteria multi-drug resistant bacteria or drug resistant bacteria. If
because,
they are, yes, Arpit.
Sir, because they like got resistant to that drug.
How? I think they were cloned to their parents. And their parents never got resistance. How did they got resistance?
So there was like change in DNA sequence.
How? Asexual reproduction is photocopying of the DNA. How did that change happen? Answer.
Mutation.
Exactly. Mutation is sudden and random, and it can happen in all life forms, ranging from bacteria. Whoever has a DNA can have alterations in the DNA. You understand? But as I told you, these are very slow events, and most of the time, if a mutation is there, it will be detrimental, it will be lethal, and it will kill the organism. But sometimes the mutation causes some beneficial effects, and those mutations are kept preserved in that genome, and next time when the bacteria will do fission, the two daughters now will be different from 3 generations before, the bacteria which was 3 generations before. Is it clear? Slightly different. So variations can also arise in an asexually reproducing organism through mutation, not through sexual reproduction, but through mutation. It is true that asexual reproduction does not give rise to variations, but there is something that can happen on the top of asexual reproduction and on the top of sexual reproduction both, which is called mutation. Is it clear, everyone? Fatima, Sumaira, Arpit,
Yes,
and
sir.
Kulsum,
Yes, sir.
Mehraj. So write down mutations in addition to recombination. Mutations in addition to recombination. Now, recombination is what happens in sexual reproduction, right? That we just studied above in the last class. And we know that through recombination, new variations are formed, recombinants are formed. Yes. Now, mutations in addition to recombination is another phenomena in biology, which leads to variations. Now, why I discuss this with you? Write down a question. Now, this question was asked in entrance when there used to be AIMS entrance. A population of asexually reproducing organisms showed genetic variation from the ancestors. The likely reason could be option 1, recombination, option 2, mutation, option 3, both A and B is possible, option D, it is not possible.
Can we write it down?
Yes, you can write down.
Can
A
you
population of asexually reproducing organisms in that question, it was a subset of the population. A subset of the population of asexually reproducing organisms showed genetic variation from their parents or ancestors. Showed genetic variation from their parents or ancestors. The likely reason could be first, recombination might have happened. Second option, mutation might have happened. Third, either both recombination and mutation is possible to happen. Both recombination and mutation is possible to happen. And fourth option, the scenario is not possible. There cannot be variation in asexually reproducing organism. Most of the time, if you do not know that how mutation can contribute, you will choose option D, that this is not possible, right? They are clones of each other. But what is the right option? You now know. Mutations. B is also not right because there can never be recombination in asexual reproduction. Okay. It can only be mutations. Is it clear, everyone? Everyone?
Yes, sir.
Yes, sir.
Yes, sir.
Yes, very good. So now there are different types of mutations. Let me tell you about some types that have been asked. Types of DNA mutation. First can be point mutation. What is a point? What is a point, people?
At a particular location.
No. Yes.
Small.
This is a point. Okay. A point mutation is where a single base is changed. Okay. So write down. When a single base of the DNA is changed, that's called a point mutation. Okay. And then a very good example of point mutation is the disease that is sickle cell anemia. You know sickle cell anemia? It happens due to a point mutation in the gene of hemoglobin, in the sequence of that hemoglobin gene. And at one single point, one base changes to another base, and that causes a disease, sickle cell anemia. That's called a point mutation. We will discuss about sickle cell anemia in genetic disorders. Just know it here. Others are second kind categories, which is called frame shift mutation. Remember, I told you that every gene has an open reading frame going back to the second chapter.
Yes.
sir.
And we read the gene as a code of 3 bases, right?
Codons.
That's called codons. Perfect. Now, in a frame shift mutation, it happens like this. Let's say this is the normal frame, not frame, normal sequence. So let's say there is a sequence A, T, C, T, G, A, T, C, G. Okay, this is a normal sequence. And in this normal sequence, this G got deleted somehow. When it was getting copied, they missed this G, or this G was just deleted due to some abnormality. So the mutated sequence will now be, tell me,
A, T, C, T, A, T, C, G.
C, T, A, T, C, G, A. It is short, right? One frame, one base short. But now let's see, look here. Earlier, the code was like this. The frame was ATC, TGA, TCG, and then further, right? Now the frame is ATC, TAT, and CGA. Do you see that the frame here onwards shifted? This T, which was in the third codon, is now a part of the second codon. Make sense, everyone?
Yes, sir.
So due to deletion, frame can be shifted. So this is called deletion mutations. So either it is deletion mutations, but these deletion mutations should not be in the factor of 3. If 3 are deleted together, then the chances are that frame will not shift. You understand? Frame will stay same, but one amino acid is lost. But deletion mutations, most of the time, almost every time, causes frame shift. Frame can also be shifted through one more way. Can you tell me what? Either through deletion or insertion, right? Let's say in the same normal gene. Let me give you an example here on.
What do you say about like if only 1 or 2 get deleted, and if a codon get deleted, what's the difference?
So if one gets deleted, the frame shifts very, very prominently. But if 3, let's say this TGA, all these 3 gets deleted, do you think the frame will shift now? It will be ATC, TCA. The frame will have one miss, but it will not shift, right? The rest of the frame will remain the same.
Yes.
You understand? Yeah, that is what I'm saying. So one way is to shift the frame is deletion mutations. The another way is to insert something. If I insert something, let's say at this place, if there is an insertion mutation, that some single base randomly got inserted. If something can get deleted, something can also get inserted by mistake. During insertion mutation also the frame will shift, right?
So how can something be inserted like inside?
How can something be deleted?
No, deleted, it can happen accidentally, but
Insertion can also happen accidentally
from
because
where
when you
did
are
it come?
when you are replicating the DNA, these bases are coming single-handedly. So let's say there are 2 strands of DNA that opened. One strand was ATGC, ATGC, ATGC. the other strand will be complementary, right? TACG, TACG, TACG. So these two were paired when this strand was joined. So to replicate a DNA, you have to first open the DNA. You understand?
Yes.
When you open the DNA, then there is a protein called DNA replicase, DNA polymerase, which is a replicase which replicates. So DNA polymerase starts making another strand. So it will put T here, it will put A here, C here, and G here, and it will keep doing that. You understand?
Yes.
So now, and it will do the same for this strand also. There will be A, T, G, C, and so like that. So at the end of replication, you will have 1 and 2 strands from one single strand. You understand?
Yes, sir.
When it is adding base by base, it is possible that here after for the A, it should have been added T, and for this G, there should be a, for this C, there should be a G, but instead it added a A by mistake and then added a G. So the frame shifted from there onwards. You understand? That is a mistake.
Okay.
Yeah. So if
But
something can get
here-
deleted, yeah.
Insertion didn't happen, no?
Insertion happened, no? This G got inserted without any, sorry, this A got inserted without any reason.
Okay. Yeah.
There was no place for A. There was no T here to make a pairing with A, but A just got inserted in that gene.
Yeah.
Okay. So that also shifts the frame. So when mRNA will be formed from this strand, it will shift the whole frame to read, and you will make a different protein. You should have been making some other protein, but now you're making some other protein which is non-functional, and you get a disease. So insertion can also happen. Is it clear?
Yes, sir.
So these are frame shift mutations, and these are the most lethal mutations because frame shift mutations change the nature of the whole protein. So write down, frame shift mutations are the most deleterious or lethal mutations because they give rise to a completely different or non-functional protein. Because they give rise to a completely different or non-functional protein. Clear? Insertional mutation, deletion mutation, both are frame shift mutations, and point mutation, where it changes. So the frame does not shift here. Okay, everyone, is it clear?
Yes.
Everyone, Maharaj,
Sir,
Kulsum, Sumaira?
Yes.
Yes, sir.
Yes. Alvia, are you following up? How's the network? I think Alvia is not connected to audio. And how is she hearing? Alvia, are you able to hear me? Am I audible to you? You can write in the chat if I am. Are you with me? I'm not sure. Okay, so this is done. Everyone has written mutation, types, and apart from types, so all these types are not in detail in NCERT, but superficially they can be asked. And then factors. What causes mutations? Do you know? Whatever causes mutations, the factors, write down, factors causing mutations are called mutagens. They are called mutagens. Is it clear? Now this mutagen can be of two types. Either it can be a chemical mutagen, for example, nitrosourea. Right, clear. or it can be a physical mutagen. For example, this you all know. Please, UV radiation. UV radiation causes mutation. Do you know during the COVID, everything was UV sanitized? All the things, objects, if you put it in a UV chamber.
But if the melanin content in the skin is more, so will UV radiation affect the person?
Yes, the chances of UV radiation affecting the person in whose skin melanin is more is very less. That's why Africans with dark skin are least susceptible to UV radiation based mutation. But fair skinned people can still get mutations through UV radiations or DNA damage through UV radiation. Okay. Is it clear, Maharaj?
Yes.
Yeah. During the COVID, like when the peak was at peak, the first peak and the second wave was at peak, you must have seen that at various places they were UV irradiating certain things like your parcels, anything which was object, the surface of that was UV irradiated. Why?
Because
UV,
it is harmful to living organisms and bacteria.
but COVID is not caused by bacteria, no?
So viruses as well.
Sorry? Maharaj, come again?
Sir, probably viruses as well.
Yes, so basically it could also damage the DNA of the or the genetic material of the virus. Kulsum is asking, do radiations include X-rays? Yes, Kulsum, radiation as a spectrum. The one which we see is also a radiation. It's called visible spectrum of the radiation. So radiation comes from, the longest one is radio waves to gamma waves. There are X-rays, there are UV rays, there are infrared, and there are ultraviolet rays, and then there is a visible spectrum. So all of that is radiation. Radiation is basically something that can travel without a medium. Basically, it does not require a medium to travel. Okay, Kulsum? X-rays are also mutagens. Yes. But X-rays do not have that high much energy as compared to ultraviolet or gamma rays. But X-rays, if you the strength of the X-ray is optimized when we do for bone analysis, that X-ray is optimized so that even if a person has around 50 to 100 X-rays in his life, he will not get mutation or any problem due to that. But if you expose someone to a very high X-ray beam for a very long time, yes, that can affect. That can cause mutations and even can cause cancers. Okay. You know, CT scan, they put the whole human in a tunnel and the human goes in the tunnel. CT scan.
Wave.
Yes, MRI or CT scan. You know, right? The whole human goes inside a machine and then the image for that organ. So that is also very, very, it's also a radiation. So
CT
scan,
it is a very high magnetic base.
Yes.
They tell you to
So
take out any metals you're wearing.
Exactly. CT scan is not recommended or is not written by doctor unless and until it is very, very important. Like if a person is dying because of a disease, and if you can do the CT, you can actually figure out how to cure it, then you will take that risk. That risk is minimum. But if someone says that, you know, I am having headache from last 2 days, I want to have my CT, doctor will say no. Not because it's a very expensive thing, because you're just having headache. Take a Disprin, you know, you understand? No need to go on in a CT machine because it's radiation. Okay, everyone, you understand? Everything
Yes, sir.
around us is radiation, from your cell phones to your radios to your televisions to your direct to home connection to your Wi-Fi, LAN networks, hotspots, all are radiations. It just depends that what 3G, 4G, 5G, all spectrums are radiations. It just depends that it should not be in a frequency where it starts affecting the human tissues and human cells and human DNA. If it is below that power, it is good for communication. If it is above that power, then it should not be used around human. Okay? Clear, people? We are living in a world of radiation. So this is all about mutation that you have to know. Types, factors, one example of each, and what is mutation. Okay, so next thing I will just discuss in brief. I will not start it in detail. Next thing that I'm going to teach you is the concepts of genetic disorders. So we will be taking on genetic disorders. Now, disorders are of two types. Okay. Either Mendelian disorders, or non-Mendelian disorders. Mendelian disorders are whose inheritance follows the laws of Mendel. And because Mendel talked about genes, right? Genes and alleles. So any genetic disorder which happens due to mutation or problem or defect in the gene is a Mendelian disorder. Okay. So Mendelian disorder is at the level of genes and alleles. Non-Mendelian disorder is at the level of chromosomes. Is it clear, everyone? For example, trisomy of chromosome 13, which is called Down syndrome. Someone told me, right? Chromosome 21. Trisomy of chromosome 21, 13, all these are chromosomal disorders. They are non-Mendelian because their inheritance is not like, it does not follow the rules of Mendel's because it's not in the gene. The problem is with the copy number of the chromosome. Is it clear, people?
Yes, sir.
Yes, sir.
Yeah. And to study both of, like to study Mendelian disorders, basically, this one, the most important thing that is done is pedigree analysis. So pedigree analysis, yes, tell me.
Only Mendelian disorders can be by pedigree analysis studied.
Yes, because it has Mendelian inheritance. So when we make a pedigree analysis, we see the inheritance. Non-Mendelian disorders differs slightly, differs in their inheritance because it's the trisomy. And most of the time, these are very, very lethal or detrimental changes. So there is no point of inheritance. So a kid, a child who got Down syndrome will not grow normally and marry a woman to produce kids. So there is no point of inheritance in Down syndrome or Tau syndrome or Klinefelter or Turner syndrome. They are mostly infertile or they have very gross deformities that they do not survive till that age. Do you understand?
Yes, sir.
So pedigree analysis is mostly done for the Mendelian disorders, where we study how this particular gene or this trait or this disorder or this disease is being carried in the next generation. So pedigree analysis is very simple concept. You all have made family trees in your junior classes, right? Your grandparents, then your father, mother, your maternal grandparents, your paternal grandparents, then you make an arrow connecting it to you and you are connected to your siblings, right? As a holiday homework. We have all done this. Yeah. Pedigree analysis is a family tree only, more biologically aligned with medical histories represented through some symbols. So you can write down pedigree analysis helps in analyzing the inheritance, helps in analyzing the inheritance of a particular trait or disease through several generations of a family. through several generations of a family. Next point. Representative symbols are used. Representative symbols are used to denote normal carrier or affected individuals. Okay. So in pedigree analysis, we use some symbols, and through that symbols, we can get to know the genetic makeup if we can study that chart properly. So studying pedigree analysis may take some time in the beginning, but once you learn it, it is the most fruitful area
Easiest.
where you can actually get and fetch marks, a lot of marks. And in entrance and also in boards, they do ask from pedigree, because that is the most conceptual thing you can ask about genetic disorders. In entrances, in the next class, I will start with pedigree analysis of some most common genetic disorders, Mendelian disorders. And then we will also solve in the class previous year's questions that have come on pedigree analysis. Okay. So
Okay, sir.
pedigree analysis and chromosomal disorder is the last thing. So we will try to finish this chapter as fast we can. So probably next weekend, this chapter will be finished with all the practice and previous year's questions, and we'll go to chapter 6. Is it okay, everyone? So your homework is you have to go back and read about pedigree analysis. What are the symbols that we used? Try to understand it on your own. If you can even understand 10-20%, that will be a add-on bonus before you come to the next class. Okay.
Yes, sir.
Yeah. So if you have any questions, I'll take the questions now before I finish class. Any questions? Yes, Fatima.
Yeah, like people say that when a mother is pregnant, that time you should like make the mother like whatever you want in the sun, like make the mother do that. Does that
Make the mother hear good things, make the mother see something,
Yeah.
like
and like
so they always put the father's
Study.
big photo in the house.
I
Yes.
don't know about that, but then like study and read.
One of my aunts had Amitabh Bachchan's photo. She wanted a kid like Amitabh Bachchan. Yes, tell me. What is your question?
So study and stuff. Does that actually affect the fetus?
No and yes, both. Let me tell you what affects and what does not. It's true that traumas and disorders, stress, distress, if you make the mother feel bad, if you make the mother feel stressed, if you make the mother feel fatigued, that will affect the development of the child. Yes. But that's not true that if you make a mother see something good, then the child will be, if you make the mother eat only white things like rice, then the child will be fair. All these are, so there is myths associated with some actual truths as well, which are being researched. Kids actually do learn when they are in the womb. That has been proven. They actually, I think I have told you, they actually show dream-like brain impulses, brain signals, which means an unborn baby, an unborn embryo is actually dreaming. But what is the brain dreaming about is a mystery, because the brain has not seen anything. No, has it seen anything in the world?
No.
Eyes are closed. It has not seen anything. There is dark inside the womb. And what is the brain dreaming about? But the point is, when we dream, we do not just see things. We also experience feelings, emotions, fear, and good things, and we also hear, right? So in the womb, right from the second trimester, the baby starts experiencing, whether it is like movements, or it is when if a mother is distressed or malnourished, the brain development will be affected similarly. So that will cause the baby to have some inborn issues, behavioral issues, or some inborn defects in psychology. And it has been proved in mouse. So if you separate a kid very early, okay, from the mother, that causes a trauma for that kid or for an offspring. So if you take the mouse pups and you separate them from the mother, early separation causes stress and depression in their minds. And if you keep doing this, and then they grow up and then they produce offsprings, and if they during the pregnancy, if the mother is given foot shock or is constantly living under fear or stress, then the baby right from like the womb starts experiencing bad things. You understand? So that can affect. But to an extent where they say that always look at good things, hear good things, listen to good music, eat only white things so that a fair baby is born. So we have to actually differentiate the myths from what is possible. Okay.
So, like, if the mother is studying anything, it doesn't, affect the.
No. If a mother is doing PhD, the baby will not be intelligent when it will be born with at least an MSc degree. No.
No, but at least a little bit.
BSc degree? No. You're saying, okay, 5th class to pass kar hi lega wo paida hote. No. Okay, you understand? So that is a myth. Okay, everyone.
Yes, sir.
Good discussions. Thank you so much, and see you in the next class. Please come prepared. We'll have more interesting discussions when we'll come to genetic disorders, and because they are also a very good area to score marks and to understand biological genetical concepts.
So, sir, then what does I didn't understand. Then, like, if this doesn't go, then what goes actually? Just only
Stress.
like if only
See,
stress?
trauma, because studies have been done on trauma. They say that a healthy mother gives rise to a healthy baby. A healthy mother, both mentally and physically, will give rise to a healthy baby mentally and physically. The chances are there. A stressed mother, a depressed mother, or a traumatized mother who has been traumatized or who have been mistreated or malnourished will give rise to a baby who will right from the birth will have psychological defects, or will have fears, or will have some personality problems.
Only on that, only stress.
This has been proved. Like in science, we never say that. Like the question that you asked, it was a direct question. So you have to ask direct pinpointed questions. For that, you say that if a mother is very learned, if she has learned 5 books and knows everything about, let's say, cosmos and black holes, she has done a PhD on black holes. Will the kid, when the kid will be born, will the kid have an inclination towards black holes? No.
Yeah.
Okay.
Yeah.
Because that kind of learning is not there. Genetic learning happens actually. Genetic, epigenetic, and some conditional learning, but not the intellectual learning. That cannot be passed on. At least not now. You never know. We can just download data and put it in someone's brain in coming future. So all you have to do to learn driving is, you don't have to learn driving. You have to just download driving module and put it in your brain.